Hello and welcome to another episode of Max Planck Florida's Neurotransmissions. I'm Lesley Colgan and I'm here with Joe. Hi, Joe.
Hi, Lesley. How's it going?
It's going great. How are you?
I'm great.
And today we are thrilled to have Dr. Tim Ryan, who is professor of biochemistry at Weill Cornell Medical and also a tri-institutional professor at Rockefeller, Sloan Kettering, and Cornell, as well as an HHMI Janelia Scholar. Dr. Ryan has really been a leader in our understanding of presynaptic function in a really quantitative manner, making incredible contributions both in technology development, which allow us to really understand things like quantal neurotransmitter release and synaptic vesicle recycling. And more recently, he's been focused on our understanding of neuroenergetics in the brain. We are thrilled to have you on the podcast. Thank you so much for joining us.
My pleasure.
Yes, welcome. And your talk today at MPFI was amazing. I say this as somebody who's not a molecular biologist, but somebody who was just witnessing just a really beautiful story that I was actually able to follow at the level of molecules doing cool stuff in the brain that's important for our lives. Just to sort of orient our audience to the nature of the type of work that you're doing in your lab, we are, dependent on energy to function. Like we have to acquire nutrients into our body that is a source that's going to power us from the molecular level to give rise to all of our amazing cognitive abilities, behavioral abilities, perceptual abilities. What happens, say I pick up an apple off the shelf at home, I'm hungry, I'm feeling some kind of internal state that's driving me to acquire food. What happens when I eat that apple that allows the composition of that apple to turn into something that my brain can use for supporting its various functions?
Right, so that's digestion.
Right.
So food sources are ultimately turned in largely into glucose through a pretty complex set of mechanisms because you can make glucose from your liver, but you can also break down their sugars in fruit. The acute thing is they go into your bloodstream and it makes the glucose go up, and our bodies have learned not to let it go too high, so we have a pancreas that secretes insulin. So we try to keep the levels steady. So the idea and the philosophy has always been the reason we like to keep it steady is because the brain needs a constant supply. So we don't want it to let it fluctuate too much.
And so when glucose in the bloodstream reaches the brain, how does that translate into something that like proteins can use to, say, do the types of actions they need to do to, if it's a membrane-bound, transporter of something, like do the physical actions that it takes to maybe move dopamine from one side of the membrane to the other?
Right. So there's an interesting bunch of plumbing that connects the bloodstream to what floats around neurons in the brain, and that's partly called the blood-brain barrier. And so there's plumbing there that's specifically meant to allow glucose to get into the extracellular space. It's pretty complicated, not fully understood. It's a frontier area, but ultimately nutrients that are in the bloodstream will get filtered into the brain through, as I said, a sort of complex set of plumbing. And then in that extracellular space, there are specialized transporters that take up things like glucose into various brain cells, including neurons. And then once glucose enters, it's an energy source because you can ultimately turn that rich carbon source, you can oxidize it. So when you turn your stove on, and if you have a gas stove, that combustion happens really quickly and gets turned into heat. In cells, that combustion is much slower, but it ultimately turned into useful things that you can do work with. And instead of generating heat rapidly, you generate molecules that you could then use to run reactions. And one of those molecules is ATP, adenosine triphosphate. So glucose goes through a bunch of steps to be turned into ATP.
Right.
Yeah, and if I understand, I think many of us have heard that the brain is actually particularly energy demanding, something like 2% of our body mass, but uses about 20% of our glucose. I felt actually very validated during your talk this morning because whenever I'm working on something difficult, like writing a grant or something, I get the munchies. I want to eat. And you actually gave some examples to suggest that that metabolic drive, or at least your metabolic state, might actually be directly connected to sort of cognitive function. I wonder if you could share a few of those examples with us.
Well, I think, as I mentioned in my seminar, I quoted work from maybe 20 years ago where people were studying in humans their ability to do a recall of words that were given. And they were given a list of 25 words, but before giving them a list of 25 words and asked them to repeat it back, they were given either a dose of an artificial sweetener or given a dose of glucose. And then they were tested on various timescales to recall those words. And the people that get glucose in these studies did about twice as well as the ones who didn't. And this was a big difference, right? And I think people have, in some ways it's shocking in some ways to people, but at the same time, everyone's had the experience you've had, which is, having a candy bar briefly certainly helps, right? And what I've encountered in the reason people are surprised, they somehow think that isn't a thing that limits brain function, right? I've even heard people say they don't think that energetics really is something that would limit brain function. Obviously, it didn't work well; it could cause dysfunction. But I think that kind of our own experience and that kind of data, that simple experiment of glucose as aspartame suggests, no, it probably is always something that's limiting how well we can do what we need to do.
Yeah, and I guess, from an evolutionary perspective, one thing that's kind of curious about the brain is that there's not like an abundant storage mechanism for energy. Like you need to kind of distribute it through the body, like get it rapidly into the bloodstream. I guess the blood is the supply of the thing. It's not like we have fat stores that can somehow be broken down into energy or something like that.
Well, actually, that's not completely true.
Oh, okay.
And I'm free to talk about it since we have this paper also on bioRxiv, but the view that the brain doesn't burn fat stores directly is quite an old one. It really dates from experiments from almost 100 years ago. It's really from the 1930s, the first experiments. But there were elegant experiments of the day, but they had a bunch of compromised things, trying to ask the question whether if you took a bunch of brain tissue and minced it up and you gave it fat, would it ultimately create CO2 or would it use up the oxygen? That's sort of what the experiment was. And they concluded, no, it didn't, compared to something like kidney or liver. Probably the subtlety was in how the mincing happened. And we know after 100 years of sort of neuroscience reductionism that you can't take apart the brain very easily and leave it doing a lot of functional things. The breakthrough, in my opinion, came about a decade ago in recognizing a human disease mutation that they didn't know what it was. I mean, they knew what the problem was. It's a class of diseases that result largely in degradation of the cortical spinal tract, but a subset of these diseases, people also have a massive cognitive problem. So intellectual disability, ataxia is vision impairment. And one of these particular mutations turns out to have been a neuron-specific triglyceride lipase. So triglycerides are the way you store fat. You package them away, they're neutral, and they get stored in lipid droplets. So our adipose tissue that we carry around are full of these giant lipid droplets. But it turns out that we didn't think neurons, you don't see these lipid droplets in the brain. But when you block this lipase from working, they build up really quickly everywhere in neurons.
Interesting. So is there this idea then that they're constantly being used, and that's why you can't find them if you go in there?
Yes, that's our conclusion basically right now.
That's very interesting.
We think of it, and this is being tested literally as we speak, but that it's probably a backup system. So when you are fluctuating in glucose too low,
you have some stabilizer.
This is a stabilizer, exactly. And when you don't have that stabilizer, then it's really not good.
That's fascinating. So the brain potentially has these redundant mechanisms to make sure that like something like blood glucose, which can fluctuate depending on like your nutritive status or whatever, would have some amount of like backup.
I mean, another fascinating one kind of related to popular culture is the ketogenic diet. So it's used therapeutically for one particular clinical indication, which is intractable childhood epilepsies. So these are kids that are refractory to any known drugs that work. And sort of a last resort, and it works in about 40% of cases in this category, but they're put on a severe ketogenic diet, which is not easy on a kid because, no carbohydrates, no fun sugars. It's not understood very well how it works at all. But ketones, of course, are another alternate fuel. It's sort of very short carbon chain. It needs to be used in mitochondria the way fat does. And then we're trying to understand right now in my lab why is there a difference in how well you can use ketones, whether you're an excitatory neuron or inhibitory neuron, which are the the things that come out of balance in seizures, for example.
Interesting. So backing up to, you described how cognitive function and metabolism are very intimately linked. You gave this example of, sugar and glucose and aspartame and memory recall. As a scientist, like using some kind of reductionist approach, how can you go about starting to understand like how changes in glucose level impact specific functional properties at the level of, an individual neuron or the compartments of neurons that are important for, synaptic transmission, for instance? Like what are some of the experimental approaches you can take to do that?
Yeah, so we do this sort of ultimate reductionism in some ways, very cellular level things in a dish. So we can make as many good measurements and control as many things. It's the great challenge in biology. When you study humans, you can't control very much. You can make observations and correlations. The further you down go the chain, the more likely you can make a perturbation to understand causation. So we're sort of at that other end of the spectrum and how we try to do science. And so for us, it's things like blocking the ability to convert glucose into ATP is an approach we take to see what are the consequences on synaptic function. And this was a discovery we made, beginning about a decade ago, is that how incredibly sensitive nerve terminal function was to this ability to make ATP on demand. And that's what excited us, realize, there's going to be a lot of interesting things to figure out here that might be quite impactful.
Yeah, so one way you did this, if I'm not mistaken, is that you used a process to actually watch the synaptic vesicle cycle using synaptofluorine. So
Yes.
this is a pH-sensitive GFP, correct? And when the vesicles are released, you can see it light up, and when they're internalized or reacidified, you lose that fluorescence. So it's basically a way to track the synaptic vesicle cycle and neurotransmitter release. So you actually use this to sort of create a really clever assay of what I would call, or what you would call, synaptic fatigue.
Yeah, we call it synaptic endurance.
Synaptic endurance. I love that. Yeah. So can you just describe how a typical synapse works and what it would look like when you actually deprive that synapse of glucose?
Sure. So just for a little background, our brains communicate largely through chemical communication, which is that one neuron releases neurotransmitter packaged into synaptic vesicles. Our individual neurons they have long axons with many, many places they will release these synaptic vesicles, maybe 1,000, maybe a typical number. At each site that they can release it, they don't rely on just one vesicle. They rely on maybe a few dozen to 100 vesicles, each storing this. But because you're so far away from where you typically make a lot of this machinery, the cell body, they have a very green recycling system in place, so that after a vesicle releases glutamate, the cell has built a machine to recover all the components of the vesicle that it needed. It recaptures them and rebuilds a vesicle locally and refills it with neurotransmitter. So the process we use with these optical probes, a fluorine-based probe, basically allows us to follow that whole thing. When we trigger exocytosis, we can watch exocytosis happen. And when the recycling happens, we also get to monitor that. And what we discovered using this assay is that if we simply took away glucose just for a few minutes and replaced it with a version of glucose that couldn't be metabolized, we broke this vesicle cycle.
So is that suggesting that really we don't, very quickly, our synapses kind of fatigue without the ongoing supply of glucose?
Yeah. That's the implication.
So, this implies that there's a certain amount of vulnerability where, if your blood glucose levels are changing, there's going to be changes in synaptic function that, you can characterize pretty well at this approach. Are there conditions that naturally happen, like maybe where you're already in a compromised state potentially with respect to neuronal function, where this gets compounded in some kind of way? I mean, you've talked a little bit about aging, neurological disease. What are some of the ways in which those problems interact with, and natural occurring problems interact with this phenomenon?
Well, sort of aging is sort of front and center on our minds because it is the biggest risk factor for explicit neurodegenerative disease like Parkinson's and Alzheimer's and other dementias. And, the world struggles to understand and develop therapies for this. And I've been viewing those through the lens of this bioenergetic balance and realizing that this incredible sensitivity to small metabolic changes might be reflected in neurodegenerative disease, that it may be the precursor, that as we age, that gets a little bit worse, trying to maintain this function well. We may have a big enough safety factor, so largely you stay cognitively intact for a long time. But then maybe one extra thing, like, so for example, I know, and I'm not a clinician, so I don't want to speak out of step, but one of the additional risk factors to age is some kind of traumatic brain injury really influences the likelihood of getting future dementia. So it could be that as you age, if you age gracefully and you hadn't had injuries, you have enough safety factor to get by. But if there had been some extra set of events, and it could be a traumatic brain injury or could be a genetic lesion you didn't know you had, that now suddenly that safety factor gets much smaller, and suddenly now you're in a position when you're 82 years old, when now you're slipping into this thing where the synapses aren't working as much.
So like naturally then you start to think about, if you were to use these types of models, like in conjunction with the reductionist approach you have, you might be able to start to identify molecular mechanisms to impart some sort of resilience to that circuit. So how would you go about doing that? Like, I mean, there's a probably unfathomable number of compounds you could potentially test. Like, how do you start to narrow your focus onto targets for that will not only, potentially lead to some kind of understanding of how you can mitigate the effects of this create resilience to this vulnerability, but also tell you something about the molecular mechanisms of how neurons and the body coexist anyway.
Right. So this same synaptic endurance assay we developed was really with the point of, could we try and make a synapse, basically I call it like making the synapse go to the gym. So we make it do a lot of exercise, but then we can, since it's in a dish, we can decide how much food to give it, how much glucose to allow it to do the work. So we picked a condition so it struggled to do this exercise, and we had the readout with this fluorine was very clear. It couldn't really do the job very well. And then we carried out what geneticists would call a suppressor screen, which is it's very cherry-picked. We didn't do a large set of genes. We just went in from what we knew about the biochemistry of converting glucose into ATP, the process of glycolysis, and said, let's try and see if there's a single one of those 10 enzymes that if one of them overexpressed could now make the system work better. And remarkably, we discovered that the 6th enzyme of the 10-enzyme cascade was the magic bullet. And a modest change in how much of that enzyme is there made a tremendous difference in how well a synapse could run with almost no fuel.
So how do you change the amount of that enzyme? So have you called this PKG1?
PGK1.
PGK1, sorry. When you express this, more than it normally would occur, you see that it kind of helps attenuate the
fatigue.
Synaptic fatigue.
Yeah.
How do you actually deliver this to the neuron?
So there are a bunch of techniques over the years, and we've used a few. You could build a virus that carries this payload and put it into cells. Truthfully, we use a simpler approach than that, which is an old thing called calcium phosphate mediated transfection, but it doesn't really matter which way you do that part of it.
Now, I think you mentioned that there was actually some hints in the existing literature that PGK1 might actually be an important specific target, which I found really interesting. Maybe you could describe some of those for our audience.
Sure. So the story began in the literature about a decade ago when a group named by a gentleman named Lesley Liu, who I don't know personally, but he led a group to trying to see if they could see if there were drugs that humans take that would actually protect the organism he studied, which were the fruit fly Drosophila, where they were explicitly trying to kill cells in the fly generically, not just neurons, everywhere, by turning on a cell death pathway. And so before turning on the cell death pathway, they said, let's try feeding the flies drugs that humans take, and maybe there's one that will keep cells from dying. And it sounds very far-fetched that this could ever work. And I can just imagine on grant study section, this is not the kind of thing that would be well received. But amazingly, they found a drug that doubled the survival of the fly, and it was a drug that's been used in humans. And to cut to the punchline of the whole thing, they don't think it was the intended target of the drug that mattered. They think it was an off-target. And they tried to figure out what that off-target was. And oddly enough, the thing that they pulled out was the same enzyme that we identified, phosphoglycerate kinase 1, PGK1. And they think this drug is a sort of a weak activator of the enzyme, not a blocker.
It's incredible. I love sort of the happenstance
Yeah.
in that story, and then it comes back to glycolysis. And actually, there's been sort of a growing focus on neuroenergetics in situations of vulnerability and disease, like neurodegeneration. And I'm wondering if you could just first introduce us to maybe some of the ways that you as a basic neuroscientist actually study neurodegenerative disease like Parkinson's in the lab itself. How can we model those in animals and start to see if these critical points like PGK1 might actually be effective in being somewhat protective?
Right. So there's sort of two routes that people take to study, try and understand Parkinson's. One very successful set of clues have come about from the improvement in technology to do gene sequencing in the last 30 years. And so by analyzing the likelihood of disease in families and figuring out everybody, you can track down like, this cousin got this disease. The only difference between this cousin and this cousin are largely comes down to maybe this small set of genes. So you triangulate. And this has led to the identification over the last 30 years of at least 24 known genetic drivers of Parkinson's. So that's one. So then you can take those known things and ask, if I make a mutation in my animal model, in a fly, in a worm, in a mouse, what aspect of this disease gets replicated? And usually some aspect does. There's always things that aren't the same, but something will show up typically in all these systems. Like in a fly, for example, it will have trouble crawling up the wall of a vial, right? And you might say that's a poor model, but it's created a problem. In a mouse, it might be a best case, it'll look like the same thing, which is in a human, in Parkinson's, the first thing to go that we think of is the degeneration of these neurons that secrete dopamine that live in the substantia nigra but project these long axons to the striatum. And so in some cases, there are version models where that begins to happen in the mouse model as well. So you can try and take that. There's a third branch, which is our second one, which we've used, which is discovered fascinatingly about 40 years ago in hospitals in the San Francisco Bay Area, when a bunch of unrelated 25-year-olds, say, showed up with Parkinson's, which is extremely unusual because that's a very, very young age to have a bunch of people that are unrelated show up with this disease. And the doctors who figured out at the time did some sleuth work and realized there had to be an environmental exposure to explain this. And it turns out they were all drug users, and they had a contaminant in the drug supply, and they traced down where this contaminant came from and what it was. It's a molecule called MPTP, and it turns out to be a pretty bad mitochondrial poison. And so this, realizing that a mitochondrial poison would cause this in a human pretty quickly, was another sort of focus, like maybe we should be paying attention to bioenergetics from that point of view. And so there are other models where you can put in other types of mitochondrial poisons and it will cause this. In some cases, you can do it specifically in dopamine neurons. Interestingly enough, of those 24 drivers I mentioned, genetic ones, 2 of them are implicated in controlling how well mitochondria work. So that was another clue that came from genetics.
So, Lesley and I were talking before we sat down to record, and you indicated something like that when you were a college student, you heard about this MPTP exposure, that was like actually one of the big hooks for you for becoming a neuroscientist to begin with.
I was really taking an undergrad class on called Animal Models of Clinical Disorders, and this MPTP story came up, and I just found it so fascinating, and it's such an interesting story. And I think the model, what happened to these people, of course, was awful, but at least we can take a little bit of solace in that the model has been widely used and has taught us a lot about
I agree.
The disease.
I mean, I know the arguments one hears is that in reality, so the success of human genetics has led to, as I said, 24 genetic clues, right? Maybe slightly more. But in aggregate, they still probably explain only maybe 15 to 20% of all the disease. So there's, in principle, 80 or 85% of people that get Parkinson's that we don't know the cause. And so the worry was you'd say, okay, MPTP, for example, will definitely cause it, but it's unrelated to the reason my grandmother got Parkinson's, right? And of course, that's always a worry, but we should follow the leads that give us strong signals.
Sure.
And hopefully in that we will learn new things. And I think that has been the case for MPTP.
And so in the case of your own research and your colleagues' research, it seems like the approach is to basically create this like focal injury that replicates many of the dysfunction of Parkinsonian symptoms, presumably, and also attacks the part of the circuit that is relevant in most, familial or genetically caused Parkinson's diseases, which it seems like there are many. You described many of the genes that have been associated. So like there's a certain phenotype for Parkinson's disease, potentially many different causes of that. Now, when you do these types of injuries, you also talked about, related approaches like using, I think it's called 6-hydroxydopamine.
Yes.
This is a toxic compound that's taken up specifically at dopamine terminals. So when you use these types of things, how can you then begin to apply this sort of, getting back to this idea of imparting resiliency to the circuit? Like, how can you use those two types of approaches in tandem to kind of learn, like, what are the limits of being able to mitigate some of the problems that you see in neurodegenerative diseases?
Right. So we took a page out of this discovery from a decade ago about this identifying a drug that would help flies live longer. And those same people with other colleagues tried it in the 6-hydropamine model in a rat and showed that this drug would protect. We were worried that we didn't want to rely too much on the fact that they thought this drug activated PGK. That evidence was probably the weakest of all the fantastic work they did, right? But it was still, in the end, it's like, well, if you want to make a better drug, you'd kind of want to know that was true, and not that the drug might be protective, but maybe it was something else. And so to make progress, we wanted to focus on, is it really this enzyme PGK1? So what we did was to see if we could boost the expression of that gene just in the right neurons that we would give the specific poison to a little bit later, maybe a month later, and ask, did we prevent them from getting sort of degenerated by this poison? And so we did that experiment. The delivery was, again, using a virus injected, so the gene would get expressed in this part of the brain. And much to my delight, this was boosting the expression of PGK1 in these neurons really did have dramatic protection against this lesion.
Yeah, I was really impressed at sort of the level of protection it seemed to have, especially, PGK is really focused on sort of this, and please correct me if I'm incorrect, focused on this one way of generating ATP in the neuron, glycolysis. And we do have, as you've already mentioned, other ways of generating ATP in the neurons. And I wonder if you could just comment on, why you think that it was able to be as effective as it was when we have these different mechanisms.
I should point out that as hopeful as I was surprised that it worked. But second, one of the reasons that it might be the last recourse for why it might have worked is that because, for example, there are genetic links of things that go wrong with mitochondria in Parkinson's, the fact that MPTP and hydroxydopamine are known mitochondrial poisons, that they may have more complex things than that, but they are known, and they cause Parkinson's-like phenotypes. It's like, we can't hope to how can we fix a thing that got poisoned like the mitochondria? We're going to try and fix the thing that fed the mitochondria. I didn't talk about this basis, but in the conversion of glucose into ATP, it's actually, there's two chunks of biochemistry. The first is called glycolysis. It's a 10-step process. You take glucose. In the end, you get a net 2 ATPs per glucose molecule. But then you make another molecule that you feed to the mitochondria to make even more ATP, a molecule called pyruvate. If your mitochondria aren't working well, that pyruvate becomes less and less useful. And that may be what's really going on, is now you've lost this sort of central, generator. And so instead, we're going to we say, listen, can we make the backup, the minor leaguer, just do a little bit better job and see if we can get by? And that seems to be the case.
And it is opening day of the MLB season.
Not in New York. We got rained out.
Well, the Yankees are in Houston now, so we at least have one game to watch tonight. We're just about out of time, but as somebody who wants to age gracefully, and wants to be protected and resilient against many of the challenges that can probably happen. And I think a lot of people listening to this podcast, because we are naturally, existentially obsessed species. I always see, when I see talks about neurodegeneration and the mechanisms that are underlying our normal function and how those coexist in like naturally occurring pathologies like Parkinson's disease, Huntington's disease, Alzheimer's disease. I always wonder, like, when a kinase is found to be particularly useful for helping to mitigate a certain amount of vulnerability in the brain, how much hope does that give us that these sorts of things are going to be developed into something that we can take as a pill that'll help us move on? So for you as a scientist, how much, what is sort of the division of motivations behind you? Because I would imagine being incredibly thrilled to find specific compounds or patterns of gene expression or whatever that can help, really seem to rescue certain amounts of synaptic function. But you're also, doing, it's an amazing combination of like translationally relevant science and also basic science where you just kind of want to understand the molecular mechanisms of how neurons work. What is that balance like for you as a researcher? Like, are you primarily motivated by just figuring out how things work, or are you really sort of driven, personally by like wanting to provide, targets for treating these types of conditions in people?
So up until 2 years ago, I would have said the balance is 100% biological discovery, 0 worrying about translational relevance. And not because I don't want to help cure disease. I didn't feel like that was best suited for my skill set. Mm-hmm. I'm not a clinician. It's complicated. I've seen mostly failure.
Yeah.
But since we've gotten into this world, and what we didn't mention is the evidence that this drug that people take, the retrospective analysis, it's quite protective. So we have a pretty big clue that in humans there's already something that works, and it was never optimized for the thing we think matters. Mm-hmm. So now I feel like, and I may be one of the few people in the world who's convinced that that's the right combination. So I do feel that now it's a bit of the burden is on me to try and convince others that we should try to push in this direction to get the right people to figure out how we are going to develop a better drug for this. I mean, it's not necessarily now anymore in my skill set, but on the other hand, it's all in my head that I'm 100% convinced this is what should be done. So I'm a bit of an evangelist now, that trying to convince people that we should do this.
That's great.
Yeah, well, I'm hopeful. So
yeah, absolutely.
Thank you so much for joining us today. Unfortunately, we're out of time, but it was wonderful talking to you.
Very good. Thank you.
Yeah, thank you so much for joining us. As always, thank you for listening to Max Planck Florida's Neurotransmissions podcast.
If you enjoyed this episode and would like to help support the podcast, please subscribe and tell your friends.